talked about how would you look for patterns in biological sequences. Um, how would you infer specific distribution of patterns? How would you represent patterns in biological sequences? Then going from there to understanding about different sequencing technologies which generate DNA sequence data, uh, and going on to the Human Genome Project, which spawned lots of different and newer technologies that have led to the growth in the generation of biological data. We also talked about in a module about how biological data is organized in the form of databases, primary and secondary, and hybrid databases. And then in the last module, we talked about how actually you come up with how do you actually sequence a genome of an organism using sequencing technologies that were developed as a part of the Human Genome Project. So now in the next module, given that we have information from lots of uh, organisms, their genomes, we have some information about what these sequences might be doing in some of the organisms. The next natural question is to ask as to how is it that you can actually gain biological insights by comparing two sequences. So very often you would have one organism on which you will be doing experiments and trying to understand the function of different the function of their genome, and you would like to extend those findings done in one organism to another organism. And in order to do this, the only similarity that you actually at the base level have every living organism has with each other is the fact that all of them are, most of them are having a DNA as their information storage molecule. And if you were to take, if you want to compare between the organisms, you would need to come up with algorithms to compare biological sequences or the DNA or RNA or protein sequences that an organism is made up of. So in today's module, the module would be about comparing biological sequences. I'm going to give you some examples of how comparison of biological sequences gives you some new insights into new biology. I'll be discussing two examples here. Then I'm going to introduce you to the alignment problem. Earlier in the earlier module, I was talking about alignment of two DNA fragments, and I was mostly talking about exact alignments or exact overlaps. So an exact overlap, exact alignment is nothing but an overlap between two sequences, biological sequences. But here we are going to talk about a little bit more interesting kinds of overlaps, which are not exact overlaps, but have gaps in between or different kinds of modifications in between. Then I'm going to introduce you to a general problem, which I call the Chandigarh Gedi problem, which basically introduces the algorithm that we use for finding, doing the alignment of between two sequences to find the longest common subsequence. Then I'm going to introduce you to another problem, which is called the change problem, which finally comes to establishing the rules that you would be taking when you would be solving the longest common subsequence problem using dynamic programming and backtracking pointers. From going from there, we are going to go into from going from dynamic programming, we're going to go to alignment graphs, which essentially give you the exact alignment between two sequences, like an exact answer, mathematically consistent answer for that. And then I'm going to talk about two different kinds of alignments, global alignments and local alignments, and how you can actually infer different kinds of biological information from those. And at the end, I'm going to talk about slightly more complicated models of how alignment is done and what is the relevance of this. So in today's class, most likely, I think I would be able to cover the first 4 to 5 points. Okay, so we all know about antibiotics. Antibiotics are substances that kill bacteria. They there is basically they're a product of natural warfare that has been happening between bacteria and fungi. So fungi bacteria attack fungi as well. They are colonizing the same niche. So fungi produce antibiotics in order to kill the bacteria. Does anybody know what kind of substances are antibiotics? There's a question. Yeah.
Sorry for
Ma'am,
interrupting.
sir, chemicals.
Right? Slides or anything.
Oh, the slides are not visible.
No, sir.
Yes, sir, there is no slide.
Is it visible now?
Uh,
Yes, sir.
it will be visible. Yes, sir.
Okay. Yeah. Sorry. So I think there was some issues there. So these are the things that I discussed about. I'm going to talk about from sequence comparisons to biological insights, the alignment, and talking about the longest common subsequences. Then I'm going to talk about two example problems in order to conceptualize longest common subsequence problem. Then this is going to be formalized in the dynamic programming algorithm and how we can get alignment graphs. And then in the end, I'm going to talk about two different kinds of alignment problems, global and local, and then different models for doing the alignment. So again, antibiotics are substances which kill bacteria. So I have already told you the background about it. So for example, this is a loaf of bread, and on top of it, you can see this white mold, which is basically um, a fungi, a fungus, which is actually the source of most antibiotics currently that we use. So can anybody tell me what are antibiotics? What kind of molecule are antibiotics? What is the composition of antibiotics?
The antibiotics
Sir,
are
antibiotics are chemicals.
In
No, don't
many.
give me the definition. What are antibiotics? Are they RNA molecules, DNA molecules?
So there,
Are they small molecules?
there are small secondary metabolites.
They are small secondary metabolites. Okay.
Yes,
So
sir.
actually, most antibiotics are short peptides. So they are essentially short peptides, which essentially means that they are a combination of amino acids which are joined together by the peptide bond. Here is an example of an antibiotic, tyrosidine B1, produced by the bacteria Bacillus brevis. Um, and because they are short peptides, they're often called mini proteins. So here is the sequence of this antibiotic. It's 1, 2, 3, 4, about 10 amino acids long. So at the molecular level, antibiotics are peptides, and it was thought that antibiotics would essentially be antibiotics such as tyrosine B1 would be made by the central dogma process. That is, they would be coded by some DNA molecule, which would be leading to an RNA intermediate, which will get translated into a protein. So it was an interesting experiment done by Edward Tatum, who thought that, okay, let me see what happens if I inhibit the ribosome basically in this bacteria. So can somebody tell me what would happen if you inhibit the ribosome in the bacteria? Would the antibiotic still be made?
If it is being formed by central dogma, then it will not be formed.
Yeah, so what they actually found was that the production of some of the antibiotic peptides, including tyrosidines, actually continued. So they thought that, okay, there is something interesting happening there. So antibiotics are not probably made by the ribosome. So this led to other scientists working on this process, and they were able to show that antibiotics like tyrosidines are actually made using non-ribosomal peptides. So that these essentially means that it is made by the process which is not dependent on the ribosome. And the proteins which synthesize these peptides are called non-ribosomal peptide synthetases, simply because they are synthesizing. So these are huge proteins, and they essentially look something like this. They have one module or one domain which synthesizes one amino acid. So essentially, if you would imagine there is an NRP synthetase that is making a tyrosidine B1 antibiotic, it would have, since it has 10 amino acids, this NRP synthetase would have 10 domains. All right. So this is how it probably would look like. Each domain will be adding a different amino acid. So the question that at that point of time researchers were asking was, because these are three same modules and they are performing practically the same function, that is adding amino acids onto each other, onto a growing chain, they should have the same sequence, right? Because it's having the same function. But now if I show you the sequence of these the three modules in the amino acid space, do you see some similarities? By looking at it, can you see some similarities?
So by looking at it, we cannot. We have to
Right.
compare like base pair by base pair, then only.
Yeah, so let us try to
So they
compare it
have,
base pair by base pair. You see that there is a A here,
Yes, sir.
right?
There's an AFD in all three at the start.
There's an AFD at all the three in start, right? So there can be many such sort of things, right? So actually when
Sir,
you, yeah,
sir, I had a question. So, sir, these beta-lactam antibiotics, are they also short peptides?
beta lactams antibiotics, I think so. Beta lactam, uh, this is a small molecule, which is I think it's a combination of small molecule and peptides, but mostly they do have some component of peptides.
Okay.
Most antibiotics.
Sir,
I can confirm that for you. I don't know at the top of my head.
sir, like penicillin is also one kind of beta lactam. Beta lactam is a group of antibiotics, and I think penicillin is also beta lactam and antibiotic or not. I,
It's not my field, so I don't know the top of my head, but I can answer this question in the next class. Okay, so getting back, so when you are looking at the sequences, one thing is very clear that these sequences are not completely same, right? So there are some differences between these modules, and we have to basically find what these differences are and how they are similar because they are causing similar function. So now, as Dave was pointing out, that there is AFD, AFD, AFD found in all these sequences. So before even doing that, let us just align these sequences as if they were DNA fragments, right, for sequencing. And when we align them, then you see that there are three locations which are exactly the same. So these three locations are highlighted here in red. Okay. So there are these three columns which are conserved. Now, if we would like to do what Dave was suggesting, that okay, there is an AFD, AFD, AFD here. So what we need to do is we need to shift the second sequence slightly on the right side. And as a result of which you would actually now find that, okay, now the AFD columns are aligning. They're actually these two L's in between start to align, these two G's start to align, right? So there's now after the three that we were finding earlier, there are actually 8 more columns which are conserved. So just like that, you can actually add more gaps in the sequence and more alignments start to come up, more and more common sequences start to come up. All right, so now the question is, how does one do the introduction of these gaps in order to basically obtain similarity between two sequences when they are aligned along with each other, right? So earlier when we were talking about genome assembly, we were only talking about exact alignments, exact matches, exact overlaps, but here there could be overlaps with gaps between the two sequences. So this is the basic idea. The problem that we're trying to solve in this module is how do you actually achieve a good alignment. Now, what do you do before I actually solve that problem? How does it help us if we get good alignments? We essentially start to look at conserved regions or conserved columns here in these three modules. And since they are performing similar functions, so we can actually suggest that these um, conserved columns might be involved in the conserved function of these three modules. Now, because these three modules are also performing difference in the function, like there is a difference in the conserved function that each one of them is adding a different amino acid, right? So after this alignment, you can also look at the positions where there is dramatic difference between each of these uh, modules, and these can essentially be also tested. So for example, in this case, the ones which are highlighted in blue, these blue positions are actually are defining the specificity of each of these modules to add a different amino acid to the growing chain. Now this, so yeah, these are the sequences that's it. So basically by doing the alignment, you can look at similarities between parts of the same protein. The same thing can also be done across different proteins in different organisms, and then you can start to look at conserved patterns and infer some relationship between proteins or domains. Now, can somebody tell me, after the researchers would have gotten an alignment like this,
So just a
what
second.
yeah.
Could you explain the significance of the blue amino acids once again? I didn't
So
really
that's what
get
I'm
it.
coming to.
Yes,
So
sir. Please,
so
like,
these three modules are aligned here. All of them, all of these three modules are adding amino acids. So their main function is to add amino acids, to add peptides, right, to a growing chain of amino acids. But each one of these modules is adding a different amino acid. So the first module adds an aspartame. Second adds a different amino acid. Third adds a different amino acid, right? So now, as a researcher, the fact that you know that these are the locations which are conserved between these modules, but each one of these modules is also adding a different amino acid, what experiment would you do to figure out which amino acids here are responsible for doing that?
a mutation. We will mutate one by one and check which one is doing.
Exactly. Exactly. So that's what researchers did. And they found out that at these locations, when you cause mutations, these had mutation, the regions highlighted in blue, the amino acid specificity of that specific module changed. So they were able to basically define that these blue locations are important to define the specificity of each module towards each amino acid. Is this clear? So typically,
Yes, sir.
whenever these kinds of experiments are being done, you would try to align sequences, two or more sequences. You would like to look at the conserved locations, right? And within those conserved locations, you would try to mutate them to see, okay, which one of the conserved locations are responsible for the function. And now if you have two proteins which are having slightly different function, they're same class of proteins or same class of protein domains, and you want to find the specificity of their function. So you would look at those aligned amino acids or those aligned nucleotides in the protein sequence, because whose mutation leads to disruption of the difference in the function. All right, is this clear?
So I didn't get how like these LTKVG helps in adding aspartic acid or ornithine
So
value.
that is, I'm not going to go into that, how it does that. It's a structural, I mean, imagine that this particular domain folds in a three-dimensional structure, and that three-dimensional structure is essentially touching the amino acid that it adds to the growing chain of peptides using these six or these about about these 8 locations. So this is a three-dimensional, it's a protein, right? So it's a protein domain. It's going to be folded in a three-dimensional shape. And this three-dimensional shape, these locations actually touch the amino acid uh, in a specific way so that it gives you specificity. So when this particular amino acid, aspartate, comes and touches these 8 amino acids which are folded in three-dimensional space, the binding energy is reduced so that this particular domain specifically binds to this amino acid. We are going to talk about how the structure of proteins leads to their function at the last module if we have enough time. But this is the basic idea.
Fine,
Is
sir.
it kind of like an enzyme, like enzyme works? And
It
also
is
one more question, like,
it is an
can
enzyme, right? Because it is the last name is Ase. So it's doing a enzymatic function.
okay, sir. By using these non-ribosomal proteins, what is the name? Like, yes, non-ribosomal proteins. We will have only three kinds of amino acid added, or like other amino acids are also
No,
there
no.
in.
So as I showed you, if you focus there in this particular NRP synthesis, there are 10 modules. So different amino acids, different antibiotics will have different combinations of modules, different length of modules, and they would be specific to each peptide that is being made. All right.
Okay.
This
Yes, sir.
is just an example to show you how the sequence alignment works, right? And how we can actually get some functional insight on the sequences based on that. Okay.
Yes, sir.
Right. So another example of how sequence alignment helps is cystic fibrosis. Cystic fibrosis is a fatal disease. It basically mostly affects the respiratory system. You have a lot of mucus being made in the lungs, and actually 1 in 25 humans are supposed to carry the faulty gene for cystic fibrosis. So whenever two people who have the faulty gene they come together, then there is 1 in 4 chance that the baby which is born is going to have cystic fibrosis, and the life quality of life life expectancy is dramatically reduced in this case. So when researchers were looking for the cystic fibrosis gene, they in 1980s they narrowed down the fact that there were mutations happening in this in the genome of people who were specifically having cystic fibrosis in the chromosome 7, and they were looking for what genes are lying on chromosome 7, and they found that a lot of similar genes which are having ATP binding, like a lot of ATP binding proteins were found in this region, and because of this, the same idea that we showed earlier, I showed you earlier when they were comparing NRP synthetase domains. In this case, they were finding similarity between ATP binding proteins in a lot of in this specific region of cystic fibrosis gene location, and they found out that cystic fibrosis gene actually is encoded by what's called the CFTR protein, which essentially is a transmembrane protein, and it regulates sodium levels. Because this was one hint that the researchers had, that people with cystic fibrosis had abnormally high level of sodium in their sweat, so their sweat was very, very salty. So by putting all these things together, they were able to search for new sequences in basically sequences in the database which were known to have ATP binding function, and which were known to have these kinds of transmembrane protein domains, right? So by looking for these kinds of uh, sequences in the database of known database of genes, they were able to basically find the CFTR protein, and this is how it looks like. In the CFTR gene specifically, when you have the normal gene, the channel it can be regulated properly, and the sodium ions can be regulated properly, sodium and chloride ions. And but when you have the mutated version of this protein, it basically disrupts the balance, causing uh, causing the body to react by making more mucus. And there are specific enhancement of like specific mutations in this gene that they found was different in the people who had the disease, and these mutations were mapped to specific regions of the gene that are essential for its function. But now, in order to do all these things, they all started with the fact that there was some information about this a region of the genome which was heavily mutated in people who had cystic fibrosis, and in this specific region they found a lot of proteins that were ATP binding, and they found out they knew that people had the symptom of having a lot of sodium in their sweat. So as a result of which they already knew that these are the kind of things that the gene might be having. So they had already list of genes which were known to be ATP binding, and they had list of genes which were known to actually be regulators of sodium, right? And because for regulating sodium, it's an active process, so you require ATP. So they were specifically looking for those genes, and in these when they were screening for these genes that were ATP binding, they could then discover the gene for cystic fibrosis. So here is a question: since I have shown you how the Human Genome Project was done, can somebody think how they could actually pinpoint a small region that was heavily mutated or heavily different in the people who had cystic fibrosis versus people who did not have cystic fibrosis? Any ideas of how this might have been done before? So this is 1980s, right? So you do not have the ability to sequence the entire genome.
But RFLP simply can be done.
Okay, so RFLP can be done. So what would you be doing? You will be taking the DNA of the people, you will be digesting it with a restriction enzyme, and you will be comparing it between people with disease and without disease.
Yes, sir.
How would you specifically compare do the RFLP for chromosome 7 specifically? You're on the right track.
Yes, sir. There will be some specific patterns, right? In most of the cases, RFLP will have uh, different kind of a specific pattern, and when this is being a cystic fibrosis is there, there will be
You're just repeating what I'm saying.
completely different.
How do you know that that particular region is coming from chromosome 7?
So you could do some sort of karyotyping for the
Okay,
chromosome.
so you can separate chromosomes based on karyotype, and then what would you do?
So like
Karyotyping is just separating chromosomes based on length. So what's the meaning of? I did not
Yeah.
understand.
So, but you can identify chromosomes, right, based on length. So if you combine
Yes, sir.
these two techniques, so you can do karyotyping, right, to identify the chromosome. And if there are large changes in the chromosome of people who get cystic fibrosis, then you would already see the branding pattern to be different in the people who have cystic fibrosis, right? Either that would happen, or you can specifically take the chromosome 7 of people who have cystic fibrosis, a lot of chromosome 7. You can literally pick up the chromosome 7 using a pipette, right? And then you can do RFLP on those fragments. All right, so this is the kind of challenges that people were facing earlier before genome sequencing came about. That's why you have to realize that it's a really big innovation that is going to change how diseases work. So
Sir,
now
just a
I
small
give you an
question. Sir, can the banding, sir,
yeah, please ask.
sir, can this banding can be used for identifying even a specific gene? Like a banding, use different kinds of banding used for
No, it cannot be done for
extending the.
specific. No, it cannot be done for a specific gene. That's why you always talk about a gene locus here. So you say that a part of the human chromosome was changed or looked different.
Yes, sir. Okay, sir. Okay.
Yeah. So I have talked to you about CTFR, CFTR, sorry, which is basically playing a role in cystic fibrosis. Now here are certain links that I want you to search and want you to read about to find out which is the most prevalent genetically inherited disease in India. I'm going to ask you questions about it. There are three papers, not one paper, one Nature article, and one link of one website of researchers who are working on it. I want you to specifically understand what gene is involved, what kind of mutations in this gene lead to the disease, and what are the current ideas behind solving this disease. All right. So coming back to the problem, now if we were to look at the different A domains with after insertions of spaces in what's called the conserved core of this module, it's absolutely unclear, right? How do we decide where to insert spaces, right? And if we have actually achieved the best alignment out of these three sequences, because you can imagine that we can insert more spaces and can start to align these sequences in a better way so that more columns are having more amino acids, the same kind of amino acids, right? So this is basically the idea of sequence alignment. So the idea of sequence alignment is that you have been given, if you were to go back, you've been given two sequences, you have to align them together so that maximum number of letters, in this case nucleotides, are going to be matching, right? So for example, you can quickly see this TGC, ATGC, right? Here it's TGC, ATGC, right? So this is the middle part, which is most common, and this is where you want to reach algorithmically. All right. So this is what sequence alignment is. To sort of define the problem, you have been given two sequences, and you have to basically align them in such a way that you have maximum similarity between the two sequences. Earlier, when we were talking about a similar problem, I introduced you to the concept of Hamming distance. Now, Hamming distance counts mismatches between two strings rigidly, right? because Hamming distance is going to compare the ith symbol of one sequence against the ith symbol of another sequence. So if you were to look at calculate the Hamming distance between these two strings, can somebody quickly tell me what the Hamming distance would be?
So
I think it's how much?
eight, because all the positions are different.
All the positions are different, right? But we do see that there's a pattern between these sequences, right? So
Yes,
since
sir.
biological sequences can actually have insertions and deletions in them, right? That's a natural process of mutations. You do not actually expect that the ith symbol of a sequence is going to be equal to the ith symbol of another sequence. So in sequence alignment, the ith symbol of one sequence corresponds to a symbol at a completely different position. All right. And the goal is to find the most appropriate correspondence of symbols, or to enhance the number of uh, similar symbols at between the two sequences in the same sequence on the sequence, if that makes sense. So basically you can have, let us say you have these two sequences. Insert I can have an insertion here, so this gap can be inserted here and this gap can be inserted here, and you see now we have this kind of an alignment, right? Where about now if you take a look at sort of the pseudo Hamming distance of these two, you would see that the Hamming distance here is 2 between these two sequences. Similarly, if I were to look at this alignment, the Hamming distance is 3 here, right? So again, the question is, how do you do this mathematically? And how do you know that you have achieved the best possible answer, right? So this is where I will introduce you to the alignment game, and we have to come up with a way to score these alignments. This is what's called a sequence alignment. You have to come up with a way to score the sequence alignment, and we have to define this problem more formally. All right. So sequence alignment, or the alignment game, essentially is a game of maximizing the number of points. All right. So whenever there's a match, you have a higher score. Whenever there's a mismatch, you have a lower score. All right. So you have to think about maximizing the number of match symbols in two strings, right? At each turn, you can move the first symbol of the sequence left and right, and you would like to retain the position of the substring if it matches. You don't need to retain it if it doesn't match, right? So for example, A and T is all matching. Now you go to this other substring GCTCC, right? So either you basically move this string to the right side, or you move this above substring to the right side, then G and T will match. Then similarly, when you will start to look at TA and you have CC, here whatever you do left or right, nothing would happen, right? So let's start to do the alignment game. This is +1, this is +1. Now you have a mismatch, so you shift it, then you start to get a match. It's +1 +1. Now you can shift it, nothing happens. You have a mismatch, you can shift it again, nothing happens, right? So the total score that you get now is 4, right? So this is what is sequence alignment. Sequence alignment essentially is alignment of two strings, and this alignment itself can be shown in this kind of a two-row matrix, right? The first row symbols of the first sequence, and the second row is symbols of the second sequence, and you would have these dashes, which is basically called insertions or deletions, that would be interspersed. Now, what do I mean by insertion and deletions? If you are comparing these two strings and you assume that they are coming from the same ancestral string, that they are related to each other, then an insertion here with respect to this stop sequence means that, let us say this was the ancestral string and a C was inserted in this new string that came up from this ancestral string, right? This is something that can happen during the course of evolution. Now, when you talk, let's say this top one is the ancestral string, and the string below, which you have got here, actually this T was deleted. Similarly, at this location, this T was deleted. And at these locations, these two locations, the A was changed to a C. So these were the different kinds of mutations that happened that changed this ancestral or old string into this new string. All right. So in the case of Hamming distance, we are only looking at mutations, right? But here we are also, in the case of sequence alignment, we are also looking at Besides mutations, we are also looking at insertions and deletions. Is this clear to everybody? Show of hands, please. I want this thing to be absolutely clear.
Yes, sir, clear.
Sir, I had a question that how do we decide which string to move? Because the last four nucleotides, none
Yes,
of them are
Deepak, that
missing.
is what I'm going to tell. That is what I'm going to tell in the next slides. So again, just to remind you, whenever two letters, two strings, two alphabets of two strings match, they align with each other. These are called matches. Whenever there's an insertion, that is, there is a new base or nucleotide or amino acid, it could be, this is called an insertion. And if there's amino acid or nucleotide missing, these are called deletions. And these other locations are called mismatches. All right. So in this case, if you talk about matches in this alignment, so ATGT is forms their common subsequence. So the longest common subsequence problem essentially is we have to find the longest common subsequence between two strings. The input is two strings and output is the longest common subsequence of these strings. All right. So this is the formalization of the problem. Now I'll give you some examples of how do you actually sort of motivate this problem. So this is uh, the photograph of how Chandigarh looks from a plane, right? Chandigarh is the capital of Haryana and Punjab. Can somebody tell me what this is? This is just a side note. Anybody?
Mohali Cricket Stadium.
Yeah, this is the Mohali Cricket Stadium. So there is something very peculiar about Chandigarh. Chandigarh is one of the most, I mean, it's peculiar for its architecture because the city was a planned city. In the idea that you can actually be in any part of the city and it looks like practically the same, right? So some people actually get bored of it. But this gives rise to an interesting observation that actually all the streets and are like in this kind of crisscross pattern. So for example, if I were to take this particular row, right, this actually represents a road like this, and horizontal roads can be represented like this. All right. So in this sense, Chandigarh is actually like many planned cities of the world. It's like Barcelona or Manhattan, who also have this kind of a crisscross pattern. So this pattern actually I took from Manhattan. All right. And within a city like Chandigarh, whenever you have these roads crisscrossing between the intersection of many roads, you would actually have lots of sites that you would want to see. All right. So basically, this entire city can be made into a grid like graph of crisscrossing roads. Right. So now, if you want to go from one part of the city to another part of the city, right? And the question is, while you are going from one part of the city to the other part of the city, you want to maximize the number of intervening sites, right? Or intervening, I don't know, intervening sites that you would want to see when you walk from one part of the city to the other part of the city, right? So the place where you start from is called the source. The place where you end up is called the sink, right? So this can actually be looked at in this context. I sort of simplify it. This is your source and this is your sink. And every time you actually look at a site, you actually give yourself a score while you are going in this, traversing in this maze. All right, so this is how it specifically looks like. You can actually now start to represent this entire thing as a matrix. And the number of sites that you will be able to see uh, is equivalent to the weight of these nodes. So the node that you start at 0, 0 is basically weighted 0. And then from 0, 0, when you go to 0, 1, so 1, sorry, 0, 1, this is the first column. Let us say you had 3 sites that you were seeing here, right? Then from example from 0, 1, if you go to 1, 1, there was no site that you could see between these two nodes, right? Between these two streets. So you get a weight 0. So you can represent the entire city of Chandigarh in this kind of a graph. Now, the interesting problem that a lot of people it's actually quite popular in Chandigarh is the problem of wasting time. That is also called the like basically going in a car and making songs about this route, which is basically trying to find the longest path in a rectangular grid of the city, right? And while you are on this path, you would want to see maximum sights, right? On you want to eat somewhere and things like that, right? So that's what I sort of call the Chandigarh Gedi route problem. So Gedi essentially means that you are going in a car and going around the city, wasting time. So the input here is a weighted rectangle, right? Grid, like I showed you. And the output is the longest path that you can take from the source, that is your starting position, to the sink in the grid, which is the position where you end up at. All right. And again, you can actually see a lot of popular culture around it. So now, if you would want to solve this problem, one algorithm that we have already talked about earlier that can be used is the greedy algorithm, right? So can somebody tell me how would the greedy algorithm work here?
It would choose the path which have maximum score
Which is the
at
maximum
the
score,
current.
right? So let us say we are at 0 here. So maximum score is 3, right? Now at this point, the maximum score is 2. This point, maximum score is 4. Then it is 4, right? At this point, the next one is we can choose any of these 2 or 3. You can already see the choice of the path that you make actually depends, it directly influences what is the next path that you're going to take, right? So let us say we take this 2, right? Then we are going to be getting a 1 in the next, right? Next road. But if we take this 2, we are going to get a 3. So let us say this is easier for us to do visually. So we take a 3 here, right? And after we have taken a 3, the next path that we're going to take is going to be a 1, and there is going to be a 3, right? So this is going to be your path. All right. And each time you take this path, you are going to increase the score, right? And 23 is going to be the score that you're going to be achieving in this kind of a greedy algorithm. Do you think this is the maximum score? This is the maximum number of sites that you can see while you're traveling Chandigarh, or when you're traveling this kind of a graph?
No,
No,
so the
because
score would be max.
we could get another score if we travel by another path. As in, if we take 2, then 5, then 2, we could get 24. So
Right.
this.
I can just also do another greedy path where I have this graph, right? And whenever I start from the path which has most number of heavily weighted edges, heavily weighted edges, right? So there is 6, there is 7, there is 5, there is 8. So if I basically go like this, right? Already there is 2, 2, 8, 12, 17, 24, 30, 33, 34. I can actually just get a 34, right? So how do we actually do this? So greedy algorithm doesn't work here that well, right? So now again, what do we have to do? We have a directed graph trying to formalize the problem further. We have to find the longest path between two nodes in an edge weighted directed graph. So remember, edge weighted means that each edge has a different weight. So this can be akin to the problem that I was showing earlier in the De Bruijn graph, where the number of fragments that are forming the node actually represent the weight of that edge, right? So different edge weights can be like that. So similarly, the different edge weights here is, yeah, in this case, the number of sites that you can see on that road, right? The input is an edge weighted directed graph with the source and sink nodes, and the output is the longest path from the source to the sink in this directed graph. Okay, everybody's clear on this?
Yes,
Yes,
sir.
sir.
All right. So now let's connect the two things between the alignment game and finding this kind of a route in a matrix. So if I were to basically say that whenever I have a match, I am going to move in the diagonal direction. Whenever I have a gap, whenever I have an insertion, I'm going to move eastwards, right, on the right side. Whenever there is a deletion, I'm going to move south, with downwards, right? And even if it's a mismatch, I'm going to move diagonally uh, along the along the matrix, right? So let us say these are the two alignments that we already had. Now let us represent these alignments in the form of a matrix like this, right? So let us say this is the first sequence that we originally had, and this is the second sequence that we originally had. So now let us see. So whenever there is a match, we move diagonally. So we move diagonally, right? Like this. Then there is an insertion, so we move right, right? So there is an insertion, so we move right here. So now there is again, we move diagonally, G and T, because G and T are exactly the same. Then there is a deletion. So should we move right or should we move left?
Down.
We should move down, right? Then we move right, or then we move diagonally, then we move down again, right? So now what I was showing you earlier as the problem for reaching from one point to the other point in a kind of a graph, weighted graph, this also represents a path of an alignment. All right. So now if we were to maximize the score of such an alignment, right, then we would basically be mathematically solving the problem of getting the best alignment. All right. So there can be many alignment parts like this. So can somebody tell me what does this alignment path show? Represent? So there are two gaps in the beginning. The
Yes.
GTTA is matching to ATC, right? Then there is a gap here, and then TCC is mapping to ATA, right? All right. So a path in this kind of a representation of the two strings is akin to an alignment. All right. The highest scoring alignment is equal to the longest path in such a route. All right. So now the goal is to build a route for the alignment game and the longest common subsequence problem. All right. Here the red edges correspond to what's called diagonals, areas which have exact matches. So for example, whenever you have an A and A in the two sequences, you are going to have a red diagonal here. So there is an AA in the two sequences here, AA. There's a TTT in 4 locations. There's a G in 2 locations, one location each. There's a T again here. There are no C's common. There are no C's, A's, G's or T's common. No commonality here. All right. So you can already populate your graph like this. So now this is where we have to sort of introduce to the next problem, which is the change problem. So change problem is also similar to what this alignment problem, and it gives us some clues of how to think about solving this problem in using some algorithms. So the change problem is that you have to find the minimum number of coins which are needed to make change, right? So the input is an integer money, right? And an array of positive integers which represent the denominations of coins that you have, right? Like 10 rupee coin, 5 rupee coin, 1 rupee coin. And output is the minimum number of coins with the denominations, right? Of those minimum number of coins that you would want to have, because nobody likes to have change, right? So now this is an example of a greedy algorithm. To solve this problem, you get money, right? Currently, you have no coins with you. So while you have money, right, you start with the largest denomination of the coin and you hope that it does not exceed the money. And then you basically add that coin's denomination into your change value and you subtract your money and then you go back. Then you again pick the largest denomination. You put it in change, you go back, you do this until you have all the money, right? But we all know that this is not a real solution because I'm quite sure most of you would have gone to supermarkets, even for example, the stores that are right there in IIT as you will be coming. You will be basically having a situation very often something similar to this, where the shopkeeper would want to give you 5 toffees instead of actually giving you the money, instead of giving you the right uh, change, right? So let us say you have 40 rupees and let's assume that in the very near future, we are going to actually have a 25 rupee coin. And these are the denominations which are available. So very often you will end up with 5 toffees because the shopkeeper is not going to employ the best algorithm, is going to employ the greedy algorithm, which is works for both him and it's not good for you. So you'll start with the maximum denomination, which is 25. The remaining is 15. Then you'll go for the 10 rupee coin. And then at the end, you're going to end up with 5 toffees. And you know that the right solution actually would be giving you 2 20 rupee notes instead of these coins and toffees, right? So this everybody is clear with this, why this is a problem.
Yes, sir.
Yes,
Yes,
sir.
sir.
that everybody faces, right? So the right way to actually solve this problem is to ask the question recursively. So here I'm going to introduce you to recursive algorithms. The idea is that let's say you have denominations 6, 5, and 1, and you want to make change for 9 rupees. All right. So let us say you have these are the different kinds of money denominations that you have, and this is the minimum number of coins, right? That you would need to have for each of these to make change for each of these. Now the question is you have to write a function which basically says that the function says give me the minimum number of coins in denomination 6, 5, and 1 for the coin 9, right? So now when you look at this particular strategy, greedy strategy always looks for starting with the highest denomination and then going down, right? But the optimal strategy basically suggests that you don't have to start with the highest denomination. You have to possibly explore all denominations, right? So now the way to do it is recursively, and I show you how. So what you do essentially is you don't know whether taking 6, 5, or 1, which one is going to give you the best combination of coins, right? So you basically now if you go 1, then you have to go at 8. If you go at 5, you have to go look for 4. If you go at first coin is 6, then you have to go for 3, right? So that's how you will move. On the other hand, what you can do is you can actually look at all these possibilities, right? So what you say is the minimum number of coins that I will be requiring to give for the change for 9 is going to be the minimum of the minimum number of coins if I take 6 as first choice, minimum number of coins if I take 5 as first choice, minimum number of coins if I take 1 as first choice, right? Because these are the 3 denominations available, right? So minimum number of coins for 9 is equal to the minimum number of coins for 3, 4, 8, and because you have already chosen 6, 5, or 1 plus 1, right? Similarly, minimum number of coins would be now for 3 would again correspond to all the possibilities for minimum number of coins for 3, for 4, and for 8, right? So you keep on recursing, you assume all possibilities, and you keep on sort of going down the possibilities. You explore all these possibilities like that. So recursively, minimum number of coins for 3, you're going to be doing the same way. Let us say for 3, you will have 3 coins, right? Because you can only have 1. For 4, you'll have 4 coins. For 8, again, you will start to go down this path, right? So when you have talk about recursive money change, you actually write this function recursive money. Money is 0. If the money is 0, you return whatever you had. You have minimum number of coins is infinity because that's where you start from. If your money is greater than the coin denomination that you have, you add that into the number of coins and you call this recursively. So within this recursive function, you keep on calling the same function again and again. All right. You call the function within the function. So this is a recursive function. So in order to solve this, there was an interesting idea that was put forth by Richard Bellman. He basically suggested the idea is whenever you are solving this problem, why don't you actually start to solve the problem for all the possible combinations beforehand, right? Rather than actually going recursively backwards, right? Because when we are doing this, when we're exploring the answer for 9, we are actually essentially exploring the answer for all the intermediate steps, right? So when you'll go for 3, you'll have to solve for 1, you'll have to solve for 2, you'll have to solve for 1, right? When you'll solve for 8, then you'll have to solve for 7, you'll have to solve for 6, you'll have to solve for 5, right? All these different kinds of cases. So what Richard Bellman suggested is that let us, instead of recursively calling the functions, which takes a lot of time computationally, let us just solve the minimum coins problem for all the possibilities. All right, so how does he do that? He suggests that let me give you the minimum number of coins for 0, which is how much? 0,
Zero.
right? Minimum number of coins for 1 is 1, for 2 is 2, for 3 is 3, for 4 now is actually 4, for 5 is actually 1, because, right, you can just directly go for 5. For 6, now based on 6, you just have to add a 1 here. So it's going to be 6, right? Directly you can have 6. For 7 is going to be 6 and 1. For 8, is going to be 6 and 2, right? You will have 2 steps, 7 and 1. And for 9, it's going to be basically 1 step from this 3, right? So basically you end up calculating all the intermediate possibilities. And this is a faster operation because these are matrix operations rather than recursive operations. Recursive operations are memory intensive. All right. So when you want to basically find the correct change, the dynamic programming algorithm version of this is basically you calculate all the intermediate values, that would be requiring. And based on these values, you then start to basically see whether the updation of your previous value versus the next value, you don't have to start greedy, but you have to start from the best value that you got before, right? So this is the algorithm for that. Again, you start with infinity. If your m here is greater than coins, you keep on doing this, right? And you are going to now populate the entire matrix with the values. So please take a look at this. And we are going to talk about how we actually use this approach to solve the alignment problem using dynamic programming in the next class. So just to give you a heads up, dynamic programming has nothing to do with programming. This word programming was just added by this researcher in order to fool the people who are paying money to do the research, because that during that time it was 1950s, and computer research was basically getting funded left, right, and center. Something currently like this is happening in biology as well. All right, so in the next class, I'm going to talk about how dynamic programming and backtracking pointers can help you solve the longest common subsequence problem. And I'm going to introduce then how this leads to the alignment graph. So very often these problems are also called the Manhattan tourist or Manhattan problem. I just change it to Chandigarh so that it can relate it to more. And two kinds of alignments, global and local alignments, and how we can actually change these things and get apply different models to extract more biologically meaningful information. I'll stop here and take quick questions. If there are no questions, you are free to leave for the next class.